So, as the name of title is New Drug Discovery and Development, so from this indicates that we are dealing with the new entity, new molecules which are meant or management for the pharmaceutical aspects or for the management of disease. So let's proceed for the further. What are the various parts? What are the various consequences? What are the various issues which might be observed, which might be faced during the, we can say that, drug discovery. In the past, most drugs have been discovered either by identifying the active ingredients from traditional medicines or by discovery by the chance. So, from this statement, we have observed two types of discoveries are there. First is by obtaining their API from the traditional medicines. We can say that by using a particular API from the medicament or from the, we can say that if we talk about the various pharmaceuticals as well as pharmacological aspects. Second is by the by chance discovery. By chance discovery mean to say that if we talk about the penicillin, this is the one of the best example that is discovered by the Alexander Fleming. But now we know diseases are controlled at molecular and physiological levels. So it is also a part of it. In the previous, the diseases are different, but nowadays, as we can say that upgrading with the material things, but at the same time, the things are also there. The type of disease that might be also differ from one to another. Also, shape of and molecules at atomic will is well understood. In the nowadays, what we can say that we have various options. We can predict the drug activity. We can evaluate their activity. We can potentiate the drug moiety so that it can work in a better way or a better manner. Next is the drug development is process of bringing a new pharmaceutical drug to the market once a lead compound has been identified through the processing of the drug discovery. So let it take example of the same how these things are going on. For example, we have already some entity for the cancer, but we want to improve their pharmacological action by reducing their side effect. So, what we can do, for example, if we take example, we know if a particular amount of that groups are available. For example, methyl group was present at a particular cyclic ring. So, for the same, if they are responsible for the side effect, what we can do, we can also determine the QSAR, that is their qualitative structure activity relationship, which means that which ensure that which type of moiety that might be feasible for the pharmacological action that without, we can say that it's suffering any kind of pharmacological action. Mean to say that the effect of that drug might be not reduced, but we are trying to reduce the we can say that side effects which are harmful for the moiety. So for the same, what we can do, for example, we are trying to take A and B in case of a pharmaceutical or pharmaceutical chemical basis, and we want to product obtained in the form of C. Now we don't know at which concentration that particular moiety give their particular action. So for the same, different trials are there at a different concentration. So for the same, if we talk about the initial, for example, we have connected in first 100 batches. So after the 100 batches, we can observe that we can find out that at which a definite or proportional amount of drug that gives a particular amount of result. So after the evaluation of 100 batches, we can able to say that yes, this batch is giving a particular result. For example, the content of your batch, that is your excipient A might be 20 or B might be 80 or vice versa. It might possible the B might be 20 or it might be 80. So we doesn't know about this, but after proceeding with the practical aspects, after proceeding with their evaluation, after proceeding with their clinical, we can say that clinical parameters, we can able to say that this moiety has a particular effect on the this side. So it includes preclinical research on microorganisms and animals, filling of regulatory status such as via United States as well as the FDA. Various agencies are there which are involving for the basic, we can say the research for the, we can, when we are trying to proceed with the new moiety, we have also various official standards are there for the same. We have to fulfill all the requirements for the standards given by the various official bodies. So after confirming that, whenever we able to obtain, yes, this moiety is giving a little amount value about the same. As we have discussed about the cancer moiety, if we are working on the same at after completing the 100 batches, we can say that yes, this moiety have a we can say that particular amount for the activity for the cancer site also. So for a investigation, new drug to initiate clinical trials on humans and may include the steps of obtaining regulatory approval with the new drug approach to the market of drug. So, as the topic is here, the development mean to say that from the initial means from the preparation of their API, mean to say that from the chemistry part, from the pharmacochemistry, then it might be the part of pharmacological, then the last part, that is the pharmaceutical angle, that is the major core part. So after obtaining the particular moiety, the last part of this is the pharmacology aspects. So this after this completion, after preparation of a complete market preparation, this will be available in the market in the form of a particular dosage. It either might be in the form of capsule, tablet, or we can say that other route. So it also depend upon the physiochemical property of the drug as well as the BCS classification of the drug. Yes, next is the modern drug discovery involves the identification of screening hits. Screening hits means, as we have discussed, we have prepared more than 100 batches for the AB to obtain the C. Now, from the total hundreds, we have observed that at the concentration of 20 A and 80% B, this concentration gives a particular action against the cancer. Now question came in mind here, how we can say that it has anti-cancer activity. So for the same, we have various options. We can use animals for the same. We can say that cell line studies are also there. For example, if we want to evaluate the effect of drug, for example, as we know, inflammatory markers are there, which are, we can say that in case of cancer, the concentration of that markers is increased in case of a cancer. So what we can do in this case, we can take cell line studies also. Just for example, Cacuto cell is also there, which is widely as well as frequently used by the researchers as well as for the industrial aspects to evaluate that, to confirm that how this moieties can be effective for the dose regimen. So I'm coding here Cacuto cell line. What we can do, we can put the interleukins 17 as well as others are also there, which are, we can say that they play a major role in the progression of disease. Mean to say that by providing the resources, we are trying to prepare a same environment as concerned with the body physiological, so that we can say that when the concentration of that interleukins is high in case of active cell lines, then what we can do, we have to access our moiety in that particular cell line. Mean to say that, for example, if the concentration of interleukins in the Cacoto cell line is 23 picogram per ml, for example. So this case we can say that this is the cancer phase, for example. But after providing our that particular new moiety or API, if this concentration is decreased to 23 to, for example, 17, 15, it means that there is some potential, there is some effects towards the declining the concentration of that particular interleukins. So for the same, we can say that, we can predict that, we can hypothesize that if the concentration of that particular interleukin is reduced from 23 to 17, it means that it might possible in the humans also, it might possible that that moiety may affect whole the particular part of the cell. So this is the one of the, we can say that, example by which we can identify a hit protein. Next is the medicinal chemistry aspects and optimization of those hits to increase the affinity. So, for example, if we take the example of a citizen, as we all know, citizen was frequently used, but due to their property of the sedation that was the, we can say that, a bad effect that was given by the, when we administered or take the citizen. But in nowadays, that citizen was converted into a levocitizen, mean to say that whenever we want to develop a new moieties, we are trying to focus on their best efficacy with minimum to minimum side effect, mean to say that that moiety should not cause any kind of side effect, because the person is already suffering from the disease, but when we are trying to provide a new moiety, if they're having a particular side effect, so what he or she can do, because he is suffering from the disease. But our motive, our target is to reduce, or we can say that, reduce their clinical symptom as well as the state of disease. So for the same, the we can say that safety profile of that moiety or that particular drug is our major goal or major objective. Next is the selectivity to reduce the potential of side effect. Selectivity means various types of receptors are there within the body, and with the various vital organs are also there. So depend upon the disease, depend upon the site, we affinity as well as the selectivity must be there. For example, I want to quote example. If we want to provide drug into the brain disease concern, for example, a person from the Alzheimer, in that case, what we are trying to do, levodopa as well as carbidopa are available within the market. So what the, we can say that, pharmacology as well as scientists are trying to do, they're trying to cross the blood brain barrier, so that the efficient amount of that particular levodopa as well as carbidopa that will be available at the particular brain site. But the, what is the barrier in this case? Barrier is the BBB, that is your blood brain barrier. So for the same, the researchers are also going on the work, so that their affinity, their selectivity is also increased. Now question came in mind, what will happen, what will happen if the particular formulation or drug have not proper selectivity or we can say that targetability. So I want to quote example in front of you. For example, 100 mg of drug is introduced to the patient. How much of drug? 100 mg. So if only 20% of that drug will be passed to the blood brain barrier, it means that 80% of rest of drug that is available as a unbound, there is no attachment of that drug within the particular site. It means that they are freely available within your blood site. So if it is freely available, it means that there is no need in the body, there is only requirement in the particular we can say that cross to the blood brain barrier. But this is not happen. What will happen? The basic thing is the only 20% of that drug is passed to the blood brain barrier. Now 80% is available with your body. So what will happen? It will cause unwanted side effect. We can say that dizziness might be also there, GIT disturbance might be also there. So a lot of things are there. It might depend on the moiety of that drug, they depend upon the exposure, depend upon the site as well as the availability of food in the GIT also. So various types of concerns are there in this favor. So but the thing is, my concern was, I was trying to understand the concept in front of you. The reason is the maximum target ability of drug that must be there, so that in particular G protein coupled receptors are there, mu receptors are there. So depend upon site, depend upon their prevalence as well as their particular physicochemical properties, the moiety must be available at the required site. For example, if I want to manage the disease which is concerned with the your particularly GIT. If I want to code example of ulcerative colitis, in this case we can say that your particular colon is involved. So what will happen? What is our primary objective? What's our primary goal? So for the same, we need a specific moiety, we need a particular formulation so that it can only be administered drug at a particular GAT. So this kind of the various examples are also there. It means from the whole concern, from the whole this study, it has been observed that if we want to manage a disease, so our primary thing is, our primary goal is we have to acquire, we have to ensure that drug release at a particular site. So let's proceed the further. One more words are there, efficacy as well as potency. Potency means the minimum amount of drug that you require for your pharmacological as well as your we can say that particular action for the management as well as treatment of the disease. So the drug must be potent, mean to say that they must be used in very less quantity. So if for the, if we talk about the in case of we can say that management of heart disease, so various types of potent drugs are also there by which we can manage the disease accordingly. Next is the metabolic stability to increase their half-life and oral viability. Now, before proceeding for the further, we must know, we have to know what do you mean by viability. It means that the rate and extent of drug to reach into the systemic circulation, mean to say that in how much time, how much concentration that is available at within your body site. Mean to say that if we talk about the injections have 100% bioavailability. If we talk about the tablet capsules, then pharmacokinetics parameters are there after the admission of the oral things, the absorption, distribution, metabolism, and excretion. These are the all processes are there. Mean to say that if we want to compare the drug, we can say that the time required by drug to reach the circulation, that is very less in case of a injectable, because they are directly linked to the blood supply. We can say that negligible time is required, very less time is required for to drug provide to the circulation. But in case of oral, as we all know, as we discussed in the right now, absorption, distribution, metabolism, questions are also there. So if we talk about the uncoated tablets, so at least 30 to 40 minutes are required to give a general action. So for the same, again I am discussing, depend upon the disease, it is also required. So for the same, I want to quote example. In case of angina, for example, we have a two option. First is that is your tablet, second is the sublingual. Now, as I have discussed how the situation is different in one of other case. In case of tablet, as we have discussed, it may take 30 to 40 minutes. But in case of angina, as we all know, it is a disease concerned with the heart. That is the very vital organ for the body. That is a majorly, we can say that a part of our body by which we can only survive. So in that case, if the angina attack is there, what we can do? Sublingual preparations are also there, by which for example we can take nitroglycerin. So I want to just clarify that formulation play a different role. Now the same case is also here. It depends upon the disease, depends upon the situation. In normal days we can take PCM in case of a for the paralytic action, but in case of a vital organ efficiency where we have a very less time, we have a less time because only fraction of seconds are there in case of angina attack. If you want to, if you are in we can say that you will be successful only when you provide a particular treatment or management for the patient who suffer from the angina. So for the same, we can't proceed for tablet, we have the only option for the sublingual. So next one is the, once a compound that fulfills all of these requirements that has been identified. So What is the basic criteria? Criteria is that must be free from toxicity, that having a good ability to reach the particular receptors. Third thing is their particular ADME. Their absorption must be there properly so that their metabolic will not cause any kind of general or as well as toxicological effect to the body. It will begin the process of drug development prior to the clinical trials. These all processes are done before the clinical trials to ensure that, to confirm that, to maintain the dignity of the system, to maintain the various official standards that are given by, we can say that, government norms, as well as the health agencies are also there, which are trying to access our things time by time, so that a good, or we can say that a remarkable things can be possible, but not necessarily involved computer aided drug design. So CADD is also there. What is CADD? By which we can say that, as we have discussed in the previous one, we have discussed about the screening hit. So in the screening hit we have discussed, we can in the initial, as we have taken example of 100 molecules might be there in the initial, but from their selectivity, their action, we can identify the better one for the further proceeding. So the same thing can be done by computer aided drug design, by which the software will conclude a result. As we have code example, we may take A, B to obtain a part C. So the computer, the system, the software will tell us that how much of percentage A and B might be there to obtain a results in the form of C. I am not giving a particular name of that solvent. That's why I am using code A and B. So for the same, we can say that if we want to proceed for the new moiety, first of all, we can proceed for the CADD, by which by means they can help to identify, help to access that which types of moieties that might be helpful for the particular things. As we have already target plot plan for the moiety, so by which the computer system or software that might help to feasible that that particular concentration, that particular amount of that drug that is able to do a particular measurement for the disease. Next is the modern drug discovery is thus usually a capital intensive process that involves large investment by pharmaceutical companies as well as the national government. So, dear ones, as we all know, from the 10 to 20 years are required to complete the whole process in an efficient way, so that for the same various teams are also there. R&D, that is your research development, and also there your basic team, that is in the lab workers, are also there. So we can say that a huge amount of required for the same, in the case form of billions rupees are required for the same. In the next slide, I will show to you, all of you, which type of companies are particular paying for the particular amount for the same. So that's why the not much more companies are involved in the research development. They are only trying to produce their already established product. The reason behind is it basically what will happen, it might possible that if we can say that more than 50 products are available for the clinical trials, out of 50 only one or two products that might pass the clinical trials phase, because it might possible the moiety that having a that are showing a results to the animal study, in vitro study, in vivo study, as well as the, we can say that, cell line study, but it might possible that they're not showing a result on the human side also. So we can't say that, we can't predict that it might possible the moiety which are obtained by us that might be feasible for the humans also. So these are the also kind of a issue, but for the time we have to try for the same. The companies are also trying for the new products, because as we have discussed the example with you, citizen and citizen, that was only established when you have faced any kind of issues and problems. So to resolve the issues. The R&D is working on the same, the scientists are working on same, so that we can provide a formulation or a pharmaceutical product without any kind of a side effect. So, as national governments mean to say that CSI labs, government DSTs, and other agencies are also there, which are working on the same. They are trying to provide give grants to the we can say that principal investigators or the researchers. The only aim is, only concern is to give a to for the betterment of nation, so that we can provide a good amount of that, we can say that good quality of drug, so that it might be feasible for the all. Despite advantages in technology and understanding of biological system, drug discovery is still a lengthy, expensive, difficult, and inefficient. Why we need to write these all sentences, as I have discussed with you, more than 10 to 20 years, we can say that tenure is there. So for the same, as I mentioned, the amount of the money for the same, that is also in a very big, then difficult. Why difficult? Because I have discussed a and B, you are taking A and B solvent to obtain to the C, but we don't know at which concentration that particular result might be there. It might be 100 batch, it might be 500 batch, but with the help of a system, in we can say that in advancement systems, as I have discussed about the CAD, so that software might be feasible that to identify the consideration, but it's not only feasible that, it might be only structural basis or on the basis of their function units. So for the same, it is only feasible when you apply that consideration in the lab also, with low rate of new therapeutic discovery. So, whenever there is a failure of that particular drug in that in the, we can say that nearby the clinical phase trial, so by which that company might think that due to involvement of a huge amount of money, that might possible that from the next company will not try the same for the new moiety. Next is the history of drug discovery. In case of 1990, at that time, herbal drugs was discovered. In 1970s, rise of biotechnology was there, and use of information technology was also there. In 1980s, commercialization of drug discovery and combination chemistry. In 1920 and 30s, vitamins and vaccines were discovered. In 1960s, in the breakthrough in the ecology was observed. In 1980s, automations as well as robotics system were developed. In 1940, antibiotics era was developed. In that case, most of the antibiotics were formed or produced, we can say that, and R&D boost due to the World War II. So after the World War II, there is improvement in the research and development phase, and in the 1950s, new technologies and discovery of DNA. In case of biotechnology, these things are going on, and nowadays, with the help of latest technique, latest things, the things are going on in the advanced mode or advanced phase. Next is the various, as I have discussed with you, top companies by R&D and their expenses. Companies are Novartis, that is spent 7.9 billions. You can observe. Merck is also there, that having 8.1. Then GSK 5.7, then Pfizer 9.1, then your Bayer is 2.3, then Novo, that is 1.7. So the purpose of this slide is only to concern that. So these companies have already spent lots of money for the same. They are trying to develop a new moiety, new entity for the management of disease, for the, we can say that, well-being of health of the concerned patients, so that they can also earn a good amount of money if they are trying to successful in a in discovery of a new moiety. Next is the process. First is drug discovery, second is the preclinical, then clinical, then FDA review, and finally that will be available with the clinics. As I have discussed, yeah, I was taking example of 100, but in figure we can say that more than we can say that thousands or 10,000 companies are also working on the initial site. But in the when we proceed for the preclinical, only 250 components might be there for the next stages. In the clinical trials, only five, and in the FDA, only one among of them that might be possible to complete the all requirements that are given by the government, to complete all the parameters which are required for the same for the management of disease that might possible. So that's why the process is very lengthy and is also involve large amount of money. In broader sense, drug discovery and development can be defined a therapeutic target of interest and include methodology, assay development, lead identification, characterization in vitro, formulation, and animal pharmacological studies, pharmacokinetics, and safety studies in animals and clinical trials in humans. As we have discussed in this case, what do we mean by animal pharmacology? Mean to say that the after the formation of that particular, we can say that hit compound which are showing their particular result in the cell line, what we can proceed, how things are going on, the same disease is induced in the animal. Mean to say that if we are trying to work on the cancer that is concerned with your GAT, what we can do, various types of methods are there by which we can induce the cancer in the animal with a particular protocol, as that must be approved by the particular animal ethic committee, so that there should no concern about the animals, how many animals are required, and what is the study objective, and what are the various parameters that must be clear with you already, and that must be already approved by the animal ethical committee. So for the same, how we can proceed for the animal, in case of we can say that for the new moiety approval. So first of all, we have to induce the disease. If we talk about the cancer, we have to induce, if we talk about GAT, the first of all, we should know what are the various parameters which must be there. For example, evaluation, if we talk about the evaluation, then there is a weight of that animal that must be there, their proper blood supply or blood pressure. So these all parameters must be considered before the treatment. Mean to say that we should have a all data about their content, their HB, how much particular hemoglobin is available to that particular animal, what about their pulse rate, what about their blood pressure, and all the about their stool content as well as stool, we can say that their consistency. This might be all be a we can say that a major part. So after the induction of disease, we also have to evaluate either the all parameters are altered or not. Mean to say that, for example, if the stool consistency was earlier that was in the normal, but after the induction of cancer, it might possible that due to gene disturbance, it might possible that the stool consistency might be slow, the stool might be softer. So after this all the parameter we can confirm, when we have confirmed and able to assess, now this particular animal is from the cancer. After that, we can provide a particular dose regimen to the animal by using their particular weight. So initial dose is started from the particular, we can say that in case of mg also. So from the mg, from 1 2 3 and 10 particular dosage regimens are also there, by which we can confirm that at which concentration the particular, we can say that drug is feasible to manage the disease. Now the thing is, maximum dose, lethal dose, as well as initial dose, that is also find from the whole study after confirming the whole concern. Basic research is also there, feasibility studies, program, non-clinical development, and clinical development. So this is the complete chart for the same drug discovery. In the initial, if we talk about thousands of compounds are also there. In the preclinical, only 250 compounds might be passed. In the clinical trials, only 5 compounds. And if we talk about the FDA, that is your Food Drug Administration, and after that, we can be able to prove only one of them. So it's not only one, it depends upon the compound, the moiety who is able to, we can say that, fulfill all the requirement which are concerned with the management of a disease. Mean to say that the moiety that is prepared by us, that must be fulfill all the criteria, all the requirement which are required for the particular disease management. Many aspects of drug development focus on satisfying the regulatory requirements of drug licensing authorities. These generally constitute a number of tests designed to determine the major toxicity of a novel compound prior to the human clinical use. As we are discussing here also, clinical toxicity studies mean to say that the compound that is prepared by us, that must be safe and efficient, as we have discussed already. So for the same, clinical toxicity studies are also there, which ensure that, which confirm that the drug moiety that is prepared by us, that is having a no toxicological, or we can say that a pharmacological bad effects on the human health. As I have given example in front of all of you, as induction of cancer disease, if our concern is just to cure our cancer, but not affect to the any other vital organs. For example, that particular moiety is doing alteration in any kind of heart as well as any other vital organs, then it should not be there. This kind of symptoms might be recorded, and we will try to improve their side effect by changing their concentration, by changing their efficient amount, by changing their pattern, so that a ultimate concern, ultimate goal can be achieved accordingly. It's legal. Next is, it is a legal requirement that an assessment of major organ toxicity performed, as I have discussed, and the test is also confirmed on the heart, lungs, brain, kidney, liver, as well as the GIT system. Mean to say that our only concern is, only a major task is to cure the particular cancer of that GIT. So it means that that moiety should not cause any effect to other sites that are already discussed. Lungs, brain, kidney, that must be in a normal. It should not like that. After obtaining that particular treatment, the SGOT, SGPT, and the other, we can say that enzyme levels or other parameters that are involved within the metabolic activity, that might be alter or reduce. It might possible that the concentration of that, the healthy or friendly probiotic might be reduced. So these types of things must be in the consideration. This must be considered, so that we can able to obtain a desired amount of that particular drug that are feasible and as well as used for the management of particular disease. The effect on other parts of body that might be affected by the drug. If the skin, new drugs to be delivered through the skin. Increasingly, the tests are made using in vitro methods with isolated cells, but many tests can only be made by using experimental animals to demonstrate the complex interplay of metabolism and drug exposure to the toxicity. As we are discussing, if we want to say that we are trying to introduce a new moiety that is concerned with the skin, so, if we talk about the disease psoriasis that is associated with the skin, so what will be the criteria? The drug must be available within the upper side of that particular skin. But what is the another thing that must be in the consideration? The drug should not be go within the blood circulation. So I'm repeating my words again. Drug should not be go within the blood circulation. Mean to say that, as we are dealing with a topical disease that is psoriasis. So far, the same. This is the kind of example. Drug must be available at the topical site so that the cells, the psoriatic affected cell might be cured by that particular disease. If the we want to cure the disease in the topically, that drug should not be given with the blood side, because if the drug will be available in the blood circulation, it will cause harmful effect, because our only concern is, our only motto is to manage the psoriatic site, that is only concerned with the upper part of the skin. So same thing is happen with the other disease also. Depend upon the disease, depend upon the site, our management, treatment, and our we can say that mode of formulation, that is also affected. So before proceeding further, it is necessary. It's very primarily. It's very, we can say that, a vital part. The proper study for the same is also a major thing to determine that either this formulation might work on this particular site or not. This will be only feasible when we have to proceed for the literature and the survey. Next, the part of the drug discovery is target selection, in which cellular and genetic targets are there. Genomics studies is also there. Proteomics is also there, and bioinformatics, lead discovery, in which synthesis and isolation of compound, combination chemistry, acid development, high throat screening in case of medicinal chemistry. The main part is library development. Mean to say that we are, as we discussed in the previous one, library mean to say that the moieties which are showing some results, we have to compile a particular data. As we have discussed example A and B, so far the same, this thing will be happen in this case of a library. SAR studies, structural activity relationship studies, mean to say that how this moiety is concerned with the other one. In silico screening, chemical synthesis in case of in vitro, drug affinity and selectivity, cell disease models, mechanism of action, lead candidate refinement. Then next is the preclinical trials, animal models of disease state, their behavioral studies, functional imaging, and ex vivo. And the last is your clinical trials. These things will be covered in the as the upcoming slide. So next is the your drug discovery. It is a phase during which the candidates or target of tested are selected on the basis of their pharmacological basis. First is the random screening, second is the molecule designing, third is the drug metabolites, and fourth one is the serendipity. Serendipity means by accidentally. Random means we are trying to collect the samples from the already hit molecules. Molecule designing, we are trying to find out that at molecule level which type of particle that AP is working. Next is your target selection. Target selection in drug discovery is defined as decision to focus on finding an agent with a particular biological action that is anticipated to have a therapeutic utility. Our basic target is basic motto is, as I have discussed in the previous also, to identify the molecules which are having a maximum pharmacological activity that must be able to cure the disease at a particular site, at a particular, we can say that, their concern. Target identification, to identify molecule target that are involved in the disease progression. Mean to say that, as I have discussed previously also, interleukins was involved in case of cancer. So in the different disease, different parameters are also there. What we are trying to do, we are trying to reduce that particular biomarkers which are responsible for the we can say that progression of a disease. If we identify them, we will able to reduce their concentration, their, we can say that, their particular availability, so that it can cure or we can say to manage our disease. Target validation, to prove that manipulating the molecule design can provide therapeutic benefit for the patient. Validation mean to say that we are trying to assure that by obtaining the particular hit moiety from the 100, or we can say that 400 batches, which type of that particular moiety or particular batch that is feasible, or we can say that having a good activity for the disease. The only concern is, only aim is to determine that either they are having efficient working for at a molecule level, not on superficial. Molecule level means in the cell levels in the with their fluidic environment, which are concerned for the same as inflammatory markers are also there, and another of the things are which are having a same activity for the disease. So mean to say that we are trying to find out the particular molecule target of the particular disease by providing a drug moiety, so that our major aim is, major objective is, major concern is to manage that particular disease by molecule level. This can be only feasible when you are particularly work on the target selectivity. Then target selection. In the target selection, the particular receptors are involved. So far, G protein coupled receptor are also there, enzymes are also there, hormones and factors, ion channels, nuclears and receptors. So in case of hormones, progesterone, testosterone are also there. So depend upon the, we can say that gender, it might also factor. So during the selection of that drug, it also factor that they are preparing for the which type of gender, because the availability of that particular hormones that might also affect. Then ion channels are there and nuclear receptors. So these are the various receptors that are available within our body system. So depend upon the affinity, their efficiency, the interaction studies are also there. Conducted to confirm that how much of drug is that particular bound with a particular receptor. Cellular and genetic targets involves the identification of functions of potential therapeutic drug target and its role in the disease state. Mean to say that genetics, mean to say that the genes are involved within our particular concern from a father to a their particular son. So far the same in case of a long disease which are having a long exposure within the body, there might change that the mutation might be also there. Due to this, the disease are not cured by normal things. So the genetics part also play an important role in the progression of a disease. Drugs usually act on either on cellular or genetic levels, chemicals in the body and known set targets which are believed to be associated with the disease. So, as you can observe, DNA moiety is also there. So we can say that that particular moiety, when they are active with a particular site, they give a specific pharmacological actions. Next is your genomics. Basically, it's the study of genes and their functions. Genomics aims to understand the structure of genome, including the mapping genes and sequencing of a DNA. Mapping genes means the genes responsible for a particular disease. We are trying to evaluate their particular moiety, as we have observed. You can observe this figure. Genetic disease risk is also there. Personalized medicine will be there. Genetic fingerprinting is also there, and there is a particular susceptibility. Mean to say that from a family background, it can be also found that if you may have observed that if their father is suffering from some chronic, we can say that a disease which might be autoimmune in the their son, it might possible that that same disease might be available with their son also. So it is due to the involvement of that genes that is involved for the father and son also. Next, the proteomics. It is a study of proteomes, the complete set of proteins produced by specific using a technology of large-scale protein separation and the modification. As we all know, the proteins play an important part within the body. So far, the same. And the analysis of protein, including protein, and nucleic acid and protein ligand interactions, will be most important for the target discovery. We are trying to find out for the specific disease which type of proteins are involved and how they doing their changes within the particular disease. In the next, that is a systemic high throughput separation and characterization of proteins with the biological system, target identification with Proteomics is a performed by comparing the protein expression level in normal and disease. So how we can confirm? We have to evaluate what is the basic effect in case of a normal, in case of a protein, in case of proteomics, and what are the various changes that are observed in case of a after the management of the disease. Yeah, we can say that in during the formation of that disease. So the basic involvement of protein that can be identified by this method. Next, the bioinformatics. Bioinformatics is a branch of molecular biology that involves extensive analysis of biological data using computers for the purpose of enhancing biological research. As we have discussed in the previous, CAD was there to identify the molecules. The same thing is there. That is the bioinformatics. With the help of system, with the help of computer screening, with the help of chemical entities, with the help of chemical compounds of target distribution, we are trying to identify that which type of particular moieties are also available there in case of a biology, by which we will be try to identify their targets. Bioinformatic methods are used to transform the raw sequence into meaningful information, genes and their encoded proteins. As we know, encoded proteins are there, so they are having a specific function within the body by which we are trying to identify their basic function and to compare them with the whole, can compare the entire genome of pathogenic and non-pathogenic strains of microbe and identify genes protein associated with the pathogenic using gene expression microarrays and gene chips technologies. Microarrays technology is also there, so that which can confirm that the particular type and that particular, we can say that availability of that particular protein, by which methods we can identify the protein sequencing. A single device can be used to evaluate and compare the expression of up to 20,000 genes of healthy as well as individual state of the ones. Mean to say that this is the methods, microarray and gene chips, by which we can identify a particular healthy as well as the disease, and we will able to observe what are the basic properties in the journal and what are the properties for the, we can say that which are suffering from particular disease, what are the changes, what are the mutations are there, what are the various coding that is altered in case of disease, that can be identified by the, we can say that gene chip as well as the microarray system. Next is the lead discovery. A lead compound is an organic molecule that act as a prototype around drug around which further optimization is centered and focused. As we have discussed, particular, it's a kind of a candidate selection. For example, we have opened a particular job requirement for a one person, but in the interview there is more than 50 persons are also there for the interview. Now the same thing is here, we need that person that fulfill our all requirement. In the same case, in the lead discovery, we need that particular compound which fulfill our all requirement in the form of their safety, in the form of their efficacy, in the form of their, we can say that maximum binding ability, in the form of their maximum pharmacological activity. Derivation of small molecules, moderators of protein functions, the process of transforming these into the high content and lead series. Mean to say that the moieties which are having a good affinity for the disease as well as their having a particular action against the disease that are lead for the further. So this is the last part of today's presentation, that is your synthesis and isolation. So in this suppression of mixing is also there, suppression of impurities, in vitro analysis and biosynthetic intermediates. Mean to say that after preparation these things are done. So thank you so much for watching this video. Rest this presentation has a further parts also, and the further things will be covered in the next lecture. Thank you so much for your time. Thank you.